What are nanobots?
Well, they're little robots that are within our cells. They're nano because they're smaller than micro. There are rotary motors, there are linear motors, there are all sorts of things operating within our cells. They are so sophisticated that they could not have evolved by natural processes. They scream at us intelligent design. If you unravel a string, it gets to a point where it just gets all knotted up and tangled. Well, with DNA, that tangling occurs. Now, there's a special motor which actually cuts the DNA and detangles it. And without that, life is not possible because the DNA tangles without it, and there's no way it could have evolved.
That's mind-blowing. What is your actual favourite molecular motor?
Probably the most amazing to me is...
So, Don, when we think of nanobots, we think of Tony Stark in the Marvel Universe, but actually nanobots are in our cells actually keeping us alive right now. Don, you're a scientist. What are nanobots?
So nano is smaller than micro, and micro is basically what you can see under a light microscope. So these nanobots are actually little robots or motors and things inside our cells, which are actually, you can't see with a light microscope. They're actually smaller than that. So you can see the cells with a light microscope, but you can't see these motors.
And so these motors are in cells and even smaller.
Really small, that's right, very tiny.
And are there nanobots that are essential to life? Like you can't live without them?
Now, many of these are essential to all life.
Right. What could one of these be then?
Well, one of them is ATP synthase. And ATP synthase synthesizes a chemical called ATP for short, or the long word is adenosine triphosphate. And ATP is necessary for all life. Everything that lives has ATP synthase or ATP and ATP synthase. And, our bodies can make half our body weight in ATP in a day.
So how does it work? How is it manufactured?
So ATP takes ADP, which is adenosine diphosphate, adds a phosphate. So it makes adenosine triphosphate. So in the process of joining these two things together, it puts energy into the compound, basically. So it takes a electrical current made of protons, unlike our electric motors, which are driven by electrons. And this drives the motor to spin, and during that process it actually puts that electrical energy into chemical energy in the form of ATP.
Right. And we know that ATP are rotary motors, but if they're so tiny, how can we be sure that they're rotary motors?
Yeah, that's a good question, and you can't actually see them rotating, as I said, with a light microscope. So scientists who were studying this back in the 1980s and early 90s proposed that it was probably a rotary motor. And a Japanese scientist actually did an experiment where he proved it was. And it was very clever. At the time, there were other scientists who said, it couldn't possibly be a rotary motor because how could it evolve? So,
well, it would be a very difficult thing for something to create.
A good question, So how could evolution produce a rotary motor like this? But the Japanese scientists actually attached a filament to the rotating part of the motor, and the filament had a fluorescent tag on it, which under a light microscope with ultraviolet light, it would fluoresce. And so he could actually see this actin filament rotating around, turned by the ATP synthase, and proved it was a rotary motor. And so the scientists who actually proposed this got the Nobel Prize for it. The Japanese scientist didn't get a look in, but he was the one who actually proved it was a rotary motor by doing something which you could see under a light microscope.
Before we continue, a quick heads up. Stick around to the end of the interview because we'll be giving away a free stream of our award-winning documentary, Evolution's Achilles Heels. You won't want to miss it. Now back to the interview. Well, that's so impressive that there's a rotary motor. And I mean, if we have a rotary motor, like where actually is it in our body?
Yeah, virtually every cell has these rotary motors, and they're particularly numerous in things like muscle cells, where you need lots and lots of ATP to drive the movement of your muscles. But there are many, many thousands of them in each cell, and they're located in our cells in mitochondria. In plants, they also have them in the chloroplasts, which is where the photosynthesis occurs. In bacteria, they're embedded in the inner membrane of the cell. So a bacterium just has a cell wall. It doesn't have membranes within the bacterium. So the ATP synthase is built into the cell wall.
So if you had to describe how this engine actually functioned on a very sort of nitty-gritty level, because we know it's a rotating engine, but what makes it up? Do we know?
Well, yes. I mean, the structure's fairly well known, although there's still question marks about exactly how it translates this electrical energy into chemical energy. But so you've got a stator, which is the static part, like our motors have a stator. So a static part which sort of holds it all together on the outside. And then in the middle is a rotor which rotates. And in the middle of the tube is a tube that's sort of embedded into the membrane. And it's in that tube that the protons actually flow up through the motor and sort of turn the rotor, which is inside that. It's a bit like a windmill, with the wind turning the windmill, or a water wheel with the water turning the water wheel. The protons actually turn the rotor in the middle. And then above the rotor, there's the three parts, which actually is where all the work happens with translating the ADP into ATP. And as the rotor turns, it changes the shape of each of these three parts one at a time as it turns around. And basically there's a groove which fits the ADP, adenosine triphosphate. It fits in the groove, and then the phosphate fits in also. And then as it squeezes it some way or other, it actually forces the mechanical energy generated by the proton current turning of the rotor. The mechanical energy is translated into chemical energy of the bond, which is formed with the phosphate. So the ATP is full of energy, ready to be used somewhere else in the cell.
So this actually is turning physical, like, energy into chemical energy in a bond.
Yes.
And that's then used back to be turned into some sort of electrical current.
Yeah, so that ATP can be used in all sorts of other things in the cell. So a lot of other machines which actually use ATP, a lot of enzymes use ATP to manufacture proteins, which our bodies are made of, and that's the stuff of life. Protein manufacture requires ATP.
Yeah.
Copying the DNA requires ATP. As I say, moving muscles requires ATP. Cilia, all sorts of things in our body that move require ATP.
So if something moves in our body at a
you can
molecular
be sure
level,
ATP is involved.
ATP is involved.
Yeah.
And so, as you've said, we produce about half our body's weight, did you say,
Yes.
in ATP a day?
A day, yeah.
That's a lot of ATP, and that means the motors must be working pretty fast. How fast are they spinning?
Yeah, about 9,000 revs per minute. It's about like jet engine speed.
I mean, I was driving my car here today, and
I hope you didn't try and drive at that speed.
9,000
blow up.
revs, my engine's are blowing up.
That's right.
So that's spinning at some pretty high speeds right there. So how big is this? We know it's in a cell. We know it's in the mitochondria. So how big is a single engine?
This is the mind-blowing thing. It's so small that 100,000 would sit side by side in a millimetre. So, a millimetre's like that much, like 1/25th of an inch. And 100,000 would sit side by side in a millimetre.
That's a lot of them that could fit into our entire body. So how could an entire motor, something that's as functional as it is, evolve?
Well, this is a problem for the evolutionary story, because all living things have ATP synthase, which strongly suggests that it's necessary for life, particularly when you consider how much bodies need ATP and how much every living thing uses ATP. And even some bacteria which don't use ATP synthase to make ATP, it's made by another mechanism, but then those bacteria use ATP synthase in reverse, so it can run in reverse. And those running in reverse, it generates a current of protons. So then that current of protons can be used to drive other things like the bacterial flagellum, which we can talk about in a little while.
So the idea that this motor that's part of every cell of our body is the idea that it's actually been evolved or came from sort of a random chance of an amalgamation of things is fairly low. So what is the proposed suggestion? If evolution seems like it's not going to be able to explain this, what is the proposed
There's
evolution
always
idea?
a story to try and explain things. And the story with ATP synthase comes from helicase. So helicase is another rotary motor which is used to untwist the DNA. So the DNA has to be untwisted, like the double helix of the DNA. If you're going to read it to make proteins, to make messenger RNA, which makes proteins, or if you're going to untwist it so it can be copied, for reproduction or cell division, this helicase is involved in that. So it's actually necessary for that process. It doesn't happen without helicase. So some people suggest that, the top part of the ATP synthase, the bit that takes the ATP and adds a phosphate to make ADP, just that bit of it, is similar to helicase. Therefore, one evolved from the other. ATP synthase came from helicase. But helicase requires ATP.
So we sort of got, I'd like to say the chicken and the egg problem, but one needs the other.
That's right.
And the ATP synthase, the more complex one, is what's needed to support the other one that was supposedly meant to come before it.
Yeah, well, a lot of things actually have the sort of equivalent of the top part of the ATP synthase, because that's where they take the ATP, run it in reverse to generate the energy to do what they're doing.
Right. So it's in fact more responsive from the ATP sort of being there at the same time or earlier.
Yeah, they sort of have to both be together, don't they? I mean, you can't have one without the other. So this is one of the big problems with the whole evolutionary story, the origin of life, is that there's no way you can have life without all the different parts functioning together to start with. There's no stepwise process by which you can get this. I mean, sometimes wave a magic wand of natural selection, but natural selection only works, if it works in terms of differential reproduction. In other words, something is better adapted to survive, more of them will survive to pass on their offspring. But if you don't have a cell that can reproduce, natural selection does nothing. It can't do anything. So it doesn't explain anything. And the attempt to explain the origin of these things by comparing them with other things which don't even have all the components. I mean, not one of the components of the ATP synthase could ever evolve. You can do the maths. I mean, maths shows that even if the whole universe is experiments over billions of years, it would never happen. So not even one of the components would ever arise by random arrangements of amino acids or anything like that. But to have the cell that functions, you need everything working together.
Yeah, well, I mean, this whole idea of ATP synthase and how integral it is to a cell kind of screams that there's probably some divine creator out there. If it took us however long to design an engine ourselves and however many brains working together, and there are even tinier nano ones that we can barely even see exist in all of our cells.
And these nanomotors are incredibly efficient. I mean, the ATP synthase is as close as they can measure 100% efficient.
100% efficient.
In other words, there's virtually zero friction or anything like that. It's just like makes our motors look pathetic.
Yeah, and so is there any sort of other rotary motors or something similar to that in cells or something that also could back up and say, hey, look, this once-off isn't just a fluke in, humans. There's actually more.
Yeah, there's one in bacteria that was the bacterial flagellum, and it's an amazing rotary motor, much more complex than ATP synthase, actually.
Right. I mean, ATP synthase was already sounding very complex.
Yeah, and the bacterial flagellum is about 4 times the width, a bit over 4 times the width of the ATP synthase.
Mm.
And it's also driven by a proton current, but actually in the reverse direction.
A lot of reversing and proton currents. Okay, so we have a proton current, not just ATP being used to make the proton current, but there is just a proton current?
Yeah, so the proton current in the bacterium actually drives the bacterial flagellum to spin.
Okay.
So it's a rotary motor.
And where does that proton current actually come from?
Well, that's another lot of stuff that's actually involved in generating it. There's other motors and other things involved in generating the ATP or the proton current. There's a gradient in the proton. So there's more protons outside the cell than inside the cell, and that actually drives the motor to spin.
Right. So we've got a proton current that's actually just driven by a difference in the amount of protons outside than inside.
So there's more outside than inside, which means there's a drive to go inside. That's the sort of way, and that drive actually spins the motor.
Okay, so we have a even more complex engine in bacterial flagellum than ATP synthase.
There's about 29 unique components in the bacterial flagellum.
Yeah, describe
proteins, that is.
Describe it more for me. How does it actually function then? We got 29 proteins in.
Yeah, so there's multiples of some of those. I mean, there's actually, with the filament, the flagellum itself, there's actually a few proteins which are multiplied many times to get the filament. But if you look down the bottom of the motor, there's sort of like a tubular apparatus, and that's the first thing formed. And it actually then, it's sort of an assembly apparatus that sort of takes the proteins that make the rest of it and puts them through the middle, and then they assemble on top, and then eventually the last thing that forms is the filament or the flagellum itself.
Right, and so we have a complex engine that has a complex way of forming and is pretty unexplainable already. So what is its actual purpose?
So you can imagine it uses some energy. So you've got this thing actually using energy. So what would be the purpose of having a motor unless it did something useful? That's the question. So what is it doing? So swimming in circles? So that wouldn't be a waste of energy, wouldn't it?
A lot of wasted energy there if you're swimming in circles.
So what the bacterium uses it for is to swim towards food or to swim away from toxins.
Right.
So how does it know the food's there? How does it know the toxins are there?
Yeah, how? I don't know.
Well, it has this sensor array which senses all sorts of chemicals, and then a computer, if you like, which processes information. So, for example, if it's swimming or living in a medium with sort of a good source of glucose, for example, over here, it has these sensors which says, oh, there's more glucose on this side of the cell than there is on that side of the cell, so swim in this direction. And then not only that, but it actually has a memory which says the glucose concentration is increasing, you're heading in the right direction. Or it's decreasing, which means you're swimming away from the glucose. And what happens then? In less than a quarter of a turn, it can actually change the direction of the flagellum and swim in the other direction.
Well, in less than a quarter of a turn. So how fast is it spinning to start
Well,
with?
here's another mind-boggling thing, is it's spinning at 100,000 revs per minute.
100,000 revs per minute.
And it can change direction in a quarter of a turn.
Like, we were talking with ATP synthase, we were talking like 9,000, and now you're giving me just over 11 times faster.
Yeah.
And in 1/4 of a turn.
A quarter of a turn, it can change direction.
It can change direction.
And by the way, we're talking about the bacterial flagellum in E. coli, which is considered the simplest one. There are ones in other bacteria which have gears and like a gear shift type thing. It can actually change gears and actually run faster or slower and all this sort of stuff, And a brake, and some of them got a brake, actually stop it.
I feel like I'm yet to see a car evolve in my life and do that, and yet this kind of thing is going on in our cells.
Yeah.
So,
and again, with virtually zero friction as well,
So 100 is sort of close to 100% efficiency.
Yeah,
again.
that's right.
That is mind-boggling.
It is mind-boggling.
And so we talked about how it moves towards food and away from toxins.
Yeah.
How precise is it with that? You said there was sort of like a computer that could kind of remember what was going on. Is it like, does it still sort of happen that like one will accidentally run into some toxins, or is it pretty accurate?
Yeah, some of the scientists involved in studying this talked about exquisite precision in terms of the information processing, the sampling, the measurement of the food or toxins or whatever, and feeding into the system. And then you've got between 5 and 10 flagella in each bacterium, and they're actually coordinated to actually work together.
So it's not just like one at the back of the cell is sort of pushing it, but you have multiple working to direct it in a very accurate and precise way.
That's right, yeah.
And that just the ability for that to actually happen in bacteria is mind-blowing, because that's so precise, and that sounds like someone's designed it.
Yeah.
Yet again, it sort of screams this divine creation.
It does. It is. It's far beyond anything we could contemplate designing or imagining, And there's even more. The assembly of the flagellum. So how do they control having 5 or so, 5 to 10? How do they control the number? Where do they assemble them? Is the assembly process, how does that work? I mean, if you've got all those components, it could get a bit messy if they all arrive at the same time.
Yeah, well.
So what happens,
So you'd have to start with the base and work your way up with all the rest of it.
And that's what happens. You think about building a house.
Right.
You have the roof trusses delivered first, don't you?
Probably not, because the
of
roof
course
goes
not.
up
Yeah, it's going to be crazy. And if the roof trusses get in the way, you wouldn't be able to dig the footings and put the reinforcing metal down, put the concrete in the footings, put the foundations down, put the floor down, put the walls down. Then you bring the trusses in at the end. Well, when the bacterial flagellum is assembled, it works like that. The components are delivered to the site in the order in which they're needed.
If you're enjoying this interview so far, you might be interested to know that we also produce the world's most widely read magazine on the creation-evolution debate, Creation Magazine. It's packed with beautifully illustrated articles showing how real science supports the Bible. It's designed for the whole family and is a great tool for strengthening your faith. Sign up now to receive 4 issues per year. Follow the link in the description. That's mind-blowing. And so with that being so mind-blowing, and I'm always more inclined to say, I think there's some divine creation going on here.
It's sort of rather obvious, isn't it?
Seems a little bit obvious. With that, obviously there are going to be people who don't think that's the case. And do they have some sort of explanation to talk about how this might have occurred more naturally?
Of course, there's always a story. So one attempt, probably the only attempt I know of to try and explain this, is certain pathogenic bacteria have a system to inject stuff into other cells, like toxins, and this is called an injectosome, and there's some similarities between it and the base of the bacterial flagellum that actually does the assembly process. So it actually injects the proteins and things into the tube, which then becomes all the different parts of the flagellum. So just that part is somewhat similar to that injection process, injection apparatus. And so the argument was the bacterial flagellum evolved from that. Well, even from an evolutionary point of view, that doesn't work, because when they look at all the different bacteria and how they compare, if anything, the injection process devolved from the bacterial flagellum.
Right, where it's
not the other way around.
Where the injection is a more simplistic version.
It's a much simpler, it's just a tiny part of the bacterial flagellum, if it was that. But actually there's comparison of the two actually shows that they're too different for them to be closely related like that, in the sense of one evolving from the other.
It seems like the more we learn about bacterial flagellum, the more complex it seems to become. So do you reckon it becomes even harder and harder to explain it as evolving to get there?
Yeah, exactly right. The more we know, the more difficult it gets to try and explain it by some sort of natural process. And so I have no hesitation in saying this screams at us there must be a super intelligent creator behind the design of this. I mean, there's even a computer program that actually operates within, I was talking about the assembly process and the order in which things arrive. We had an article in our Creation magazine not long back by a scientist who actually drew up the logic flow. In other words, a computer program type of thing, which actually is used by the bacterium to determine the order in which things are delivered to the site. And, like it's a logic flow diagram that most people couldn't understand. And yet this is in the bacterium. It's supposedly made itself by natural processes over, etc.
So we've got like a computer code that sort of replicates something that happens at such a nano scale.
Yeah, and it's all happening in this cell. You need a microscope to see it, and you can't. And you can see the bacterial flagellum in a light microscope, the actual filament and everything. You can see them swimming around, and but the actual mechanism of the motor is just amazing, just absolutely amazing.
Well, again, circling back to us talking about how it becomes more and more complex and harder to talk about and explain it as evolution. I've heard some evolutionists accusing creationists of using a sort of God of the gaps scapegoat, if you like, talking about how we don't really understand a lot of things and we might say that to explain it away. What would you say to that?
Yeah, so sort of the God of the gaps argument is, we don't understand it, therefore God did it. That sort of thing. And that's a really bad argument. I don't use that argument. I argue from the point of view of knowledge. Right. That is, when we look at these things we're looking at, what we know about them actually screams at us that there's a super intelligent creator behind these things. They couldn't have come about by natural processes. And that argument gets stronger with more knowledge. Whereas a God of the gaps argument disappears, the argument for God disappears when we understand it. And I'm not arguing we don't understand it, therefore God did it. I'm arguing we do understand it, therefore God did it. It's a very different argument.
And
a very powerful argument.
God did something so complex at such a scale that we can't even really comprehend.
I think he's smiling at the scientists and saying, hey, get your minds around this, guys.
Check this out. Check out what I can do.
Yeah, that's right.
That's awesome.
Yeah.
So we've spoken about two different kind of rotary engines that are so microscopic, like nano level in our cells, keeping us alive. Are there any kind of other engines or something similar that are also in cells that are really helpful? Well,
we can talk about linear motors, which don't rotate, but they actually move in a linear fashion. So,
right. Yeah, okay.
So we can talk about a kinesin, for example. And there's actually various forms of kinesin, but the first one that was discovered is a family called kinesin 1. And it actually walks around our cells delivering parcels.
So could you elaborate on that more? So it's delivering parcels. I mean, why do we have parcels in our cells anyway? And what are they walking on? They're walking? We've got walking things in our cells now.
Yeah, they've got walking things in our cells. So there's in our cells, not in bacteria, but in our cells, we have a road network of microtubules, and they're made of proteins. It's actually a very dynamic road network in the sense of it can be recycled and then built again depending on where it's needed and so on. So you can imagine travelling on the highway and the highway is appearing in front of you as you're driving, that sort of thing. That's the sort of thing we're talking about. But this road network, what happens is the kinesins actually walk along the road network delivering parcels of proteins.
Right.
So, for example, proteins are made here in the cell, but they might be destined to go to the mitochondrion. So what happens is the proteins which are destined for the mitochondrion have a label on them which say, deliver me to the mitochondrion.
Mm-hmm.
And you remember in high school, labeling a cell,
Mostly, yeah.
well, you had a nucleus, a nucleolus, and cell wall, and Golgi apparatus. You remember Golgi apparatus?
Squiggly line looking thing.
That's right, a couple of squiggly lines in the cell. You didn't know what it was or what it did. Well, what that does, we now know, it actually reads the address labels and packages up the proteins that are going to the same destination. So you can imagine with an address saying go to Melbourne on your letter, and so all the ones going to Melbourne get parcelled up in the same bag. Well, this is happening in the cell. So all the proteins going to the mitochondrion get parcelled up together, and then the kinesin comes along and grabs it, attaches itself to it, and then walks along the road network of microtubules and delivers them to the destination.
And so we have these things walking around on these little highways delivering proteins. What's the purpose of the proteins? Like, why are they actually so important?
So the mitochondria wouldn't work without the proteins. Now, you probably ate some starch for lunch. You had a sandwich with starch in the bread, and starch breaks down to glucose. When you put that in your mouth, your body said you need amylase to digest the starch. A signal went to your salivary glands, and the salivary glands manufactured amylase. How did it get into your saliva? Well, the amylase was transported by the kinesin, dumped outside your cell into the saliva. How quickly did that happen? Or within seconds.
So I've taken a bite of my BLT that I actually did have at lunch. You're right, there was bread as part of that, the starch. I take a bite, and before I even really know what's going on, there's been chemicals dumped so I can digest it.
Well, enzymes to digest the starch and make glucose out of it.
That's amazing. So how fast are they moving then?
Well, you might have seen the animation of it sort of plodding along, like this sort of thing.
Yeah.
That's quite misleading. I mean, it's sort of done at that speed so you can see what's happening. But they actually walk at 100 to 200 steps per second.
Per second?
Yeah.
So we're talking how many steps across a cell do you reckon? Or like how fast is that moving within a cell?
Looking at 125,000 steps in a millimetre, so that's how small it all is. But a cell is a fraction of a millimetre total diameter, so it doesn't take very long for the kinesin to do what it needs to do.
And get
in fact, I can illustrate it even better. Put your hand on a hot plate.
Yeah.
How long does it take to react?
No time at all, really. I've realised it's hot and I've taken my hand off very quickly.
Well, your brain is involved in that process.
Yeah.
Because your brain's processed a signal which says your hand's burning, pull your hand away.
Yeah.
And then your muscles have to react and all that, and that actually uses ATP. But your brain process, so you've got neurons in your brain, and then the electrical signal comes from your hand saying your hand's burning, but your brain has to process that. And so in the synapse between the two neurons, there's a little gap, tiny little gap. But there's chemicals had to be transmitted across that gap for that signal to be processed by your brain to tell you your hand's burning. And there are neurotransmitters, there's over 100 of those known, and things you've probably heard of dopamine and serotonin, acetylcholine, and there's about 100 of them that I know about. And depending on what happened with your nerves, depends on which neurotransmitters are dumped into the synapse to actually get the right reaction. So adrenaline might be one of these.
So yeah, if I touched it, I wouldn't feel calm. I'd feel like
that's right. You feel panic. Panic. Pull your hand away. So all this happens by kinesins. Kinesins actually have these neurotransmitters sitting there, ready to dump into the synapse. How quickly does all that happen?
Well, within a fraction of a second.
Fraction of a second. That's right.
All the way from
they're involved in this. The research on things like Alzheimer's and brain degeneration diseases, one of the things they're finding is that these tangles of proteins that interfere with the neurotransmitters being dumped into the synapse by the kinesin.
And
they actually bind to the microtubule, so so the kinesins can't walk along and dump the stuff in.
And so that's actually blocking the synapses from working.
The synapses don't work properly, and you get the symptoms of dementia. So they're really, these things are just absolutely amazing, and what they do and how important they are. That's just one of the families of kinesins, kinesin 1.
Yeah, and this sounds like it's got so many purposes. Are there any more purposes that you can think of that actually?
Yeah, so you've probably seen the pictures of a cell division under a microscope.
Yeah.
The chromosomes all line up across the middle in the spindle, and then they separate out and they pull apart and form 2 new cells. Kinesins are involved in that. But a different type of kinesin, because a kinesin 5 family they're called, and they have a walkie thing on each end. So rather than a walkie thing on one end and attached to a bag on the other, the vesicle, they've actually got a walkie thing on both ends, and so they walk on microtubules on both ends.
Right, yeah. Okay.
So they're actually involved in pulling the chromosomes together in the middle and then pulling them apart to make the new cell.
Right. And so that's a completely different kind of kinesin, but same family.
It's the same mechanism in the sense of it's got the walkie thing and it walks along the microtubules, but the microtubules attach to the chromosomes, and then they actually pull them apart or pull them together depending on which way the microtubules go.
And so kinesins are sounding more and more essential to my life.
They're absolutely essential to your life and mine, and everything which has a cell with a nucleus and chromosomes. So everything from yeast to humans actually have kinesins. Bacteria don't have them.
Bacteria don't
have them, no.
So if bacteria evolved into us supposedly, where did these kinesins come from?
Exactly. Where did they come from? How could they possibly evolve?
And not only where did a specific type of kinesin come from, where did all the versions of kinesin come from?
Well, you look at the kinesin 5 family, and how does cell division occur without them? With cells that have chromosomes. Like bacteria have a circular chromosome of DNA, but there's no chromosome structure like with proteins and things wrapping around it and all that sort of thing. So bacteria don't need kinesins to divide and reproduce, but everything from yeast to us does. And so if bacteria became yeast or something like that, how did they invent the whole chromosome replication, separation, cell division, all that stuff that depends on kinesins, and also the delivery system, with kinesin 1 delivering proteins into the right places. Our bodies can make at least 100,000 different proteins. Different cells make different proteins, possibly as many as 1 million, and we don't really quite know how many. So what would happen if those proteins were all made together and they weren't actually labelled and separated and delivered the way they want to be? They'd interfere with one another.
Well, it would be like as you said, like a postage system. If I didn't tell you where the addresses were, you'd just have to try and figure it out, and mail would go to the wrong spots.
That's right. It'd be a total mess. So they're totally essential to life as we know it, and they're not in bacteria. How do you invent a delivery system that depends on labels on the proteins, reading the labels, packaging them up, delivery system, the microtubules, so you've got a road network to deliver on, and all that has to happen properly or it doesn't work at all. It's not as if you can have a stepwise process by which you can get some sort of function and then a little bit of advantage. It has to all be functional for it to work at all. So it's quite totally beyond any natural evolutionary process of lucky accidents called mutations in bacteria to generate such a thing.
And so I've heard evolutionists speaking of natural selection, like a bit of a magic wand, saying that, it can work a little bit of a miracle. What would you say to that? Well,
it depends on mutations. It's not natural selection. The mutation is the only mechanism they have for generating the new proteins. So, for example, if you have bacteria that don't have kinesin, how do you get kinesin for the next stage of evolution? And what's the point of having kinesin? So if you have, even if you've got the kinesin, which has the actual active part of the kinesin is 350 amino acids, just the active part, not the whole lot. You do the calculations, 20 amino acids 1 over 20 is a chance of getting the first one right, 1 over 20 is a chance of getting the second one right, 1 over 20 times 350 multiplies by itself 350 times, you've got the chance of getting that sequence of the active part of the kinesin.
Which is a
Which
very,
is never
very
going to happen.
negligibly small number.
It's never going to happen. And even if you got that, what would be the purpose of it unless you had the microtubule road network system
For it to actually...
for it to happen and something for it to do that made it actually worthwhile. Otherwise, it's using up ATP for no purpose whatsoever.
Which in some sort of natural selection method would actually probably be bred out then if it's not actually serving any kind of positive purpose.
Exactly right. So if you had this part system which was wasting energy, natural selection would get rid of it.
Yeah, so it actually looks more like you would be more likely to lose any kind of information that was
any step
positive.
towards the functional system would be detrimental, and natural selection would get rid of it.
So design-wise, these engines are crazy. Like I can't even imagine trying to design one myself in some kind of large scale. What is your actual favorite molecular motor, though? We've talked about all these different ones. What's your favorite?
We've talked about some rather amazing ones, but probably the most amazing to me is one called topoisomerase, which is a big word. Sounds very complex, and it is.
Well, it does sound complex, so tell me more.
There's actually two types of topoisomerase, and these are present in all living things, bacteria, humans, everything in between. And topoisomerase has an absolutely essential function in all living things. And it's got to do with the fact that our DNA is a double helix, so it's twisted like this. So to read it, you need to untwist it.
Right, sort of like opening a book. You've got to untwist.
Well, not a book so much as you think about a rope. I've got a piece of string here. I can sort of illustrate the issue.
Would you say it's like the equivalent, though, of us opening a book and reading what's inside? You have to undo it.
That's like that with the DNA. You've got to open the book to read it. That's right. But in the process of untwisting the DNA so you can read one of the strands, imagine that's the two strands of the DNA, and we start to untwist it, and we get so far, I don't want to go any further.
Yeah.
Because it's twisted up and it's actually tangled. In fact, if we let go, see how it's actually twisted up and tangled?
Yeah, it wants to sort of retwist itself.
It wants to fold back on itself and twist up. And the DNA does that. And so what does a cell do? Because if this happened, that'd be the end. There's no reproduction. It can't reproduce the DNA. Not even bacteria can do it because it'll twist the same. So it has this topoisomerase actually fixes the problem.
And how does it fix that problem?
Well, I only talk about type 2. Type 2 is the most interesting one. But type 2 wraps itself around the DNA and actually cuts the DNA, double-stranded, and actually the twisted loop, it feeds back through the cut, and then it actually reseals the DNA, rejoins it.
Right.
And this motor, this amazing topoisomerase enzyme motor does that job. And so it's got a top part which opens, it's got a bottom part which opens, and it's got a middle section which does the cutting and joining. And it lets the two strands of the DNA in each side, cuts it, feeds it through, and then rejoins it, and then lets it out.
So that is to, in fact, negate the knots that keep
it
forming.
It detangles the DNA,
detangling it as it goes.
It's a detangling enzyme, yes.
So that's so...
Isn't that amazing?
That's so amazing because that's, in fact, so necessary for the DNA to be untangled.
There's no life without it.
And that's just casually going along and doing that every time a cell divides.
Yeah.
Wow.
And it uses ATP.
And there you go. Yet another nano machine that has to use ATP to function.
Yeah.
So if topoisomerase is so necessary for life, for a self-producing cell, how could life have even started then?
No, it couldn't. It could not have started without super intelligent design, putting things together. I mean, the things we're talking about today, clever scientists have worked out how they're working to some extent. which is just amazing science. I just love it. I love the science.
It's so fascinating.
It is so fascinating. Working out how God's creation works is just so amazing. Those scientists, clever as they are, could never, ever have dreamt of actually inventing those systems.
Well, they're so complex.
They're so complex. And they come along after the event and work out how they work to the best of our ability. And some very clever science, very clever scientists. They could not have ever dreamt of actually designing those things. And yet, sadly, strangely, some of those very scientists go on about evolution, how this evolved or that evolved, It doesn't make any sense.
Yeah, especially with how complex it is and the chances of such complexity having to come together at the same time so it doesn't lose that information is actually useful.
Yeah.
That there's such an infinitesimally small number that, it's almost... It feels like it's screaming at you in the face.
It's screaming at you. In fact, I spoke at a church in Melbourne some years ago, and a young guy came to me afterwards, and he said, I'm doing a PhD in molecular biology at the University of Melbourne. And he said, totally agree with I just said a little bit about these motors in my talk. But he said, I totally agree with what you're saying about these things. He said, I cannot understand my colleagues who study this stuff daily and can't see the hand of God.
Yeah.
So it just doesn't make any sense.
It doesn't seem to make sense. And I mean, I know I'm just a regular uni student asking a scientist a bunch of questions, but even to me, it seems like that is such an obvious answer. And I mean, it might be a difficult answer to agree with. It has so many implications that if there is intelligent design, there's an intelligent designer, and that actually means a lot for me.
Yeah, that's right. Yeah, that's the problem. I mean, the problem is that the science points to an intelligent designer far above us. Who could that be other than the creator God revealed in the Bible? There's no other option, really. So that's the reason there's so much resistance to following the evidence wherever it leads, which is towards... I mean, Romans chapter 1 says that God's attributes are seen in what he's created, so people are without excuse.
Yeah, and here with these nano machines, we see that this is so complex and has to be the hand of God, that this is almost irrefutable evidence for that.
I would say in today's world, the people with the least excuse for believing in God are people studying this stuff, the molecular biologists who are studying this stuff.
Well, delving into nanomachines with you, Don, has been such a pleasure. And I mean, I think I've learned so much. I don't think I've ever known so much about a cell, even after doing, biology in high school. So life clearly shows us that there's an intelligent creator. So it's not actually of natural processes, but instead someone who had to have a hand in this creation. What do you have to say about that? Yeah,
absolutely. These things scream at us that there must be an intelligent creator behind everything, and life has a purpose. That's one of the implications. Life has a purpose. We're not just an accident, a cosmic accident of rearranged stardust or something, but life has a purpose. If there's an intelligent creator who designed and purposed life, he did it with a purpose. It wasn't just for fun. And so we have a purpose, an eternal purpose, and that's what the Bible's about. That's what creation's about, to point us to our Creator. Not to live life as if there's no meaning and purpose, and when you die you become fertilizer. No, there's an eternal purpose to our lives.
So, Don, we have a documentary that you want to tell us about. What have you got to show us?
So it's Evolution's Achilles' Heels. There's 8 chapters, but there's 15 PhD scientists who explain why cosmic evolution, from the beginning of the universe to the origin of life and the diversity of life and the meaning and purpose of life, all covered. It's very, very entertaining, really high quality, lots of animations and on-site footage and things in it. It's divided into chapters, so you can sort of just watch a little bit at a time if you want to take it in slowly, but really highly recommended.
And this is actually a free stream with the link in the description if you guys want to go check that out. Don, thanks for your time yet again. Thank you for walking us through some of these nanomachines that are in our cells. It's genuinely just some of the most fascinating stuff I've ever heard. Thank you for all the work you do.
That's been my pleasure. It's been my pleasure. Thanks, James.